Hi students, today 12th classes in botany specification. Already this topic is going to variety of title is classified, which chapter is going as commonly called as plant tissue culture. In plant tissue culture, today which topic is going to be classified is namely called as artificial synthetic seeds. Before the entering of the title, what are the definitions of plant tissue culture techniques? Plant tissue culture is nothing but by applications of science in short-term time, they have to be produced plants without infective diseases caused by many pathogens. That is that good specifications of plant tissue culture. In plant tissue culture, how it is derived from artificial or synthetic seed. You know how the seeds it generally is derived from them, or which is responsible for producing seeds. Because for your daily activities, you have to be consumed different kinds of seeds as well as fruits, because the fruits containing seeds. How the seeds it must be present inner to the fruits. It's a happening. One of the important process is namely called as pollination and fertilization. So what is the pollination? Transfer of pollen grains from anther to stigma. It may be classified as self-pollination and cross-pollinations. After pollination, it must be going to fertilization. What is the meaning of fertilization? Fusion of male and female gametes. That is a normal progress. It's generally happening in flowering plants. So all kinds of flowering plants, how it's merged seed through the process of pollination and fertilization. By these two events, seeds are forming. What will be going to classify as now? Artificial seeds. How it is artificial? The seeds are merged without the process of pollination and fertilization. So there is no process of pollination, there is no process of fertilization, but you are merged seed by the process commonly called as plant tissue culture techniques. Okay, now come to the point of the title now. Artificial seed is also called synthetic seeds. Why it will be considered synthetic seeds? Because the seeds it's merged from human being are also called man-made. Synthetic seeds are also called synseed. Okay. How that seeds is derived, or which process going to be applicable to manufacture such a seed. So synthetic seeds, these are obtained from embryoid. Embryoid is also clonal, somoclonal embryos. What is the somoclonal embryo? What is the embryo? Embryo is nothing but, in general, fusion of male and female gametes resulting formations of zygote. The zygote develop as an embryo. That's a normal process. What is somoclonal embryo? Embryo is derived from somatic cell. What do you mean somatic cell? Body cell. So body cells are also responsible for manufacture of embryo. That's a commonly called as Embryoid are also called somoclonal embryo or somoclonal embryos. How that somoclonal embryo or embryoid is derived from where? Or what are the cell accompany them that can form as embryoid? So the embryoid or somoclonal embryo, it is derived from single cell. That single cell is derived from any part of the plant. You know that in plant they are having various kinds of parts, namely root, shoot. In shoot, different kinds of parts are there. You know, leaf, stipule, pedicel, then hairs, number of branches, yes, whatever it may be. So the formations of embryo is derived from single cell. The single cell derived from any part of the plant. After that, the single cell undergo divide, responsible producing mass of cells. So after divide from the single cell, resulting they can form as a mass of cells. In each cell or in each mass of cell, what are the components it must be present? It's contained dense cytoplasm, a larger nucleus, then enzymes, proteins, and the oils. So those are the substances that are generally present in every kind of cell. So that kinds of things, it not only occur in a single cell, in mass of cell they may be occur. In artificial seed, how it responsible produce commodity called as, there are a number of inert materials are used for the productions of artificial seed. So what they call inert? You know that the chemicality lecture. Inert means the substance will not activate either physically, chemically, and biologically. That's called inert materials. For the productions of artificial seed, different kinds of inert materials are used for productions of artificial seed. What are the inert materials are generally used for artificial seeds? Yes, can I list it here? One is called agarose gel. Second one is called sodium alginates. Those two are generally used for manufacture of artificial seeds. Next, can I discuss about what are the advantages of artificial seed? There are many advantages of artificial seed than true seeds. First of all, what are the first advantages commonly called as? Numerous artificial seed are produced at short time at any cost and low cost. So numerous artificial seeds are produced at any time as it will be available as low cost. Second thing, Artificial seeds are commonly called as it can easy to produce. It's not only easy to produce, it must be produced as genetically engineered plants. Are also known as transgenic plants. Is another aspect of productions of genetically engineered plants or transgenic plants. That plant it must be possess desirable traits. So what do you mean commonly called desirable traits? What is that? The interest of the gene. It must be available. That kinds of gene it must be present that to be considered as commonly called as desirable traits. That's the second thing. Third thing, it is easy to test about genotypic plants. So you know what is the meaning commonly called as genotype? Genetically plants or gene. So if you are responsible for test, it can possible to very easy what type of gene if it is present or not. That must be assessed by using artificial synthetic seeds. Fourth one, again, potentiality, storing for long time by using cryo preservation method. What is the meaning of cryo preservation? That is that advanced technology that must be discussed to be later. Can I go to the fifth one things? What is that advantage of artificial seed is commonly called as? Artificial seed produced identical plants what is the last one advantage is commonly called as? Artificial seed can alter the dormancy period resulting responsible for increasing growth at short-term life cycle. So in normal plants, it cannot possible to alter the dormancy period, but artificial seeds are alter the dormancy period because it must be shorter the time. It is not only alter the dormancy period, it also increasing growth of the plant in shorter life cycle. So those are the things it responsible for advantages of artificial seeds. Next one I am going to discuss about virus-free plants. You know that virus, one of that dangerous pathogens. In normal grow on perennial plants, so what is the meaning of perennial? The plants it must be live on many years. So the normal grown on perennial plants generally affected variety of pathogens such as bacteria, fungi, mycoplasma and virus. So bacteria, fungi, mycoplasma, virus, this is commonly called as a pathogen. These pathogen, it's generally cause a variety of plants, resulting responsible for economic loss. So there is no possible to increase produce good yield or the productivity is going to be lost. How, which way to be in order to control bacteria, fungi, mycoplasma and virus? Chemical methods can be used for control bacteria, fungi and mycoplasma. So far in your home or in your villages, you have seen so many kinds of plants are there. They can affect so many kinds of insect and pest. So you have to using so many kinds of insecticide and pesticides you have to spray to kill bacteria, fungi and mycoplasma. But no one the chemical will not be controlled the multiplications of virus. So chemical method it is not applicable to control for virus production. So which way to be controlled virus? The control virus is the only method is commonly called as virus-free plants. How you have to be obtain virus-free plants? Where it must be obtained? That is the only way. Apical meristem tip culture. So apical meristem tip cultures only irresponsible produce virus free because the tip of the meristem is generally free from virus. So that's why if you are cultivated plants virus free means where it must be obtained from apical tip meristem culture because here the tissue is obtained or the leaf primordially is obtained that must be going to the culture medium. You have to be made virus free plants. There is no any alteration in this way. Okay. What are the protocol to prepare virus free plants? The first thing. Apical meristem tip with 1 or 2 leaf primordia in sterile condition from X plant. This is the first protocol. Second thing, in vitro culture in 10% of solid MS medium supplemented with growth hormone. That is second protocol. Third one, culture is maintained by or culture is maintained at 24 plus or minus 1 degree Celsius for dark condition in 3 days. After illuminated at 2400 lux. So what do you mean commonly called as lux mean? Unit of light intensity. This is the last one is commonly called as plantlet developed from meristem culture. Finally, transfer to hardening. So these are the things, protocol to preparations of virus-free plants. Can I go to the next one is commonly called as germplasm conservation. Germplasm conservation is nothing but conservations of living genetic resources. What is that? Conservation of living genetic resources such as seed and pollen and tissues of plant material. Why you should be conserve seed, pollen and tissues of plant material means the things is responsible for future use. So, relevant plant breeding are also called selective plant breeding. So selectively, if you have to be going to breeding means the seed and pollen and plant materials are it must be stored. So conservation in living condition, if you have to be stored means that is a greatly help for future researchable works. In addition, germplasm conservation resources is commonly called as it's a part of storing seed and pollen. Why you have to store seed and pollen means, or if you have to be store seed and pollen means, the seed and pollen it must be having good viability and good fertility for long time. If you stored seed and stored pollen, it must be possess good viability and good fertility for long time means that kinds of seed, that kinds of pollen, it must be used for that must of seed and pollen it can be used as crop improvement and future genetic resources. So that purpose only the seeds and pollen it must be stored in specific banks are made. That bank is commonly called as seed bank or pollen bank. That seed bank it must be store different kinds of seed. Likewise, pollen bank is responsible to store different kinds of pollen. But that kinds of pollen, that kinds of seed, it's having always good viability and good fertility. That viability and good fertility pollen and seeds that can be used as crop improvement and future genetic resources. In addition here, germplasm conservation it's also involving in gene bank and DNA bank. Gene bank, that means it can store rare genetic material, it must store be here. Which is the rare genetic material? For example, Sandalam album, sandalwood. So it's a rare plant. That is now is going to endanger nature. Such an endangered plant you have to be stored in gene bank. That the genetic material you have to be stored in DNA bank. So gene bank, DNA bank, the kinds of plant resources you have to be used to prepare allied plant breeding. You know that what even commonly called as allied superior variety. A superior plant resources you have to be made with the gene and genetic materials derived from gene bank and DNA bank. In addition here, the gene bank, DNA bank, how it must be responsible for maintenance commonly called as a good biodiversity. Because all living organism, survival to be good means really you want be good biodiversity. If you maintain good biodiversity, mean it must be maintained gene bank and DNA bank. It not only make regarding what good biodiversity, it also maintain food security of the plants. Can I go to the last one topic is commonly called as cryo preservation. Minus 190 degrees Celsius, 96 Celsius degrees. So what is the called cryo preservation? Cryo preservation is also called cryo conservation. So cryo preservation or cryo conservation is nothing but conservations of plant components such as protoplast, cells, tissues, organs, organelles, extracellular matrix, and enzymes. So what is the answer to say? Conservations of protoplast, cell, tissue, organ, organelles, extracellular matrix and enzymes. Those are subjected to conservation, or those are subjected to preservation by cooling mechanism. So the substance responsible for cooling under low temperature ranges of minus 190 degrees Celsius by using liquid nitrogen. So those material, if you have to be stored in under cooling low temperature in liquid nitrogen means that component is always present to live conditions. If not as stored in under the condition mean the storing protoplast, cell, tissue, organ, extracellular matrix and enzyme became is inactivate. In addition here, if you have to be allow extreme low temperature in more than 196 Celsius, if you have to store such a kinds of metal mean, those all things became the work can be stopped. The work only it can be stopped, but all cells are it can present to alive condition, not to be death. The work will be stopped. When that work it will be again to be continuous. After that, if you have to be contributed any research work in protoplast, cell, tissue, organ and organs mean those kinds of cell just you have to allow them room temperature. If you are allowing room temperature mean gradually the cells are from the stopped condition to come as regarding alive conditions. That's why all kinds of plant material, it must be stored in conservation during the time of dormant state. So if it is not stored as a dormant state, those kinds of material which leads to regarding death. Okay. In cryo preservation method, what are the cryoproductants which can be used, or which productant to be used, such as the productive agents such as sodium sulfoxide and glycerol or sucrose that will be added before preservation process. Or what are the material you are going to be cryo preservation, if you have to explain, those kinds of substance it must be added before the preservation process. So the sodium sulfoxide and the glycerol and the sucrose, those the things it is commonly called as cryoproductants. That cryoproductants, it is product cells and different tissues from stress of freezing temperature. If we are not using such a substance, mean those kinds of all material will be going to be death. That's why what are the plant material you have to be contributed the research, those material, everything it must be stored on under liquid nitrogen. That must maintain the temperature is commonly called as minus 196 degrees Celsius. Thank you.